when we are saying the viruses, what you should all know is viruses, the largest virus is pox virus, smallest virus is the parvovirus. Okay. Viruses, since they don't have their own cell, acellular, they're obligate intracellular. They have to come inside a cell to reproduce, right? They need a host cell for their reproduction. So is that understood? Next. What is a virus made up of is virus is made up of a structure nucleocapsid. Viruses are made up of nucleocapsid. That's the word right here for you. Nucleo means the nucleic acid like DNA or RNA. Capsid is the protein coat surrounding the nucleic acid, which is made out of proteins called as capsomers. Capsid is made out of proteins known as capsomers. Is that clear enough, doctors? So nucleocapsid. Nucleic acid is DNA or RNA. Capsid is capsomers. Capsid would be the capsomers. So nucleic acid and this thing. Okay. Now, if this much understood what we just said here, let us now continue over here. Okay. Depending on naked or envelope virus, how are you going to identify them first of all? See, everybody will have nucleic acid. You can see this part, nucleic acid is present here also. Nucleic acid, it is present here also. Yes or no? Same thing. Nucleic acid is present here also, here also. Extra thing is the envelope, that's all. An envelope is made up of glycoproteins. And you should know, the more stronger viruses between the two, we may think it is this virus. It's not. It is not a strong virus, right? Envelope virus is actually a weak virus. Which is the strong virus? Naked virus. Naked viruses are strong viruses. They are resistant to things that we have, like, alcohol. Next. Shapes of viruses, etc. You should have a look at them later, like each one of them. They are the description. Now, sabse pehle, you should all just recap this word, that is happy. Why are you supposed to know the word happy? Happy are the DNA viruses. These all viruses are the DNA viruses. Anybody else apart from the happy word has to be a RNA virus. So happy is the DNA viruses. Everybody else are the RNA viruses. Okay. Next, from the happy word, some viruses are naked, known as PAP. And you need to know papillomavirus, adenovirus, parvovirus for this. Papillomavirus, adenovirus, and then the parvovirus. Okay, so that's about this guy. Next, RNA viruses are all except happy. RNA viruses are these ones, which are all except which word? Happy word. That's the first thing. Now, what are the naked RNA viruses? You can just have a look here. Park or crab mnemonic, however you memorize. Picorna, Astro, Rio, Calici. Now, in the Rio, you can see rotavirus, which is the most common cause of diarrhea in children. Okay, it's the most common cause of diarrhea in children. Next, Calici virus, it causes diarrhea in adults, the member known as Norwalk virus. Norwalk causes diarrhea in the adult people. Okay. Hepatitis A and E belongs in this family only. Hepatitis A and E come in this one. And since majority of this family members are causing diarrhea, they're all going to spread by fecal-oral routes. Okay. So what is the route of these guys? If I ask you, you should all know it is fecal-oral route. It is going to be fecal-oral route. So this part will be revising coming up shortly. First is herpes virus, what you're looking at. Okay. First is the herpes virus, what you're looking at. You all know there are around 8 members of herpes like this. Okay. There are 8 members of herpes as such. So what you're supposed to know is the third one is called as varicella zoster. Fourth one is known as Epstein-Barr virus. Fifth one is called as the cytomegalovirus. Okay, they are just names given to some of them. So 1, 2, 6, 8, they are just numbers. The ones in middle, like 3, 4, 5, they have some names also in addition. Now, please know HSV1 and 2. HSV1 causes the lesions above the waist. Number 1 is on top. That's the hint, right? So above the waist. HSV2 causes lesions below the waist. Okay. Now, if you have understood this much, please see the point. Herpes labialis, this is the most common infection that is caused by HSV1. Okay, this is seen on the lips in the form of this ulceration. This is HSV1 lesion, HSV1. Okay, next, if they want to describe the encephalitis here, they're going to describe this word temporal lobe encephalitis. Okay, temporal lobe encephalitis. Is that understood by all of you? It produces what? Temporal lobe encephalitis. Done? Next, it remains latent in trigeminal ganglia because if it has to cause the lesions in and around the face, then this virus should live near the face only. Okay, so that's why we are saying trigeminal ganglion. Okay. HSV2, it causes lesions below the waist. And the genital herpes primarily, this is the one that's something you should be knowing. Genital herpes, this is caused by HSV2. And what is the characteristic of genital herpes? Painful recurrent lesion. Painful. There are only two things that are going to be painful. One is chancroid, another is herpes. Okay, the ulcers itself, they are only painful in two diseases in STDs. One is chancroid, another is herpes. Okay, and that exception you might have already seen called as Mollaret's meningitis. How about this image if I show you? Shingles is this unilateral rash that involves dermatomes. It's a unilateral rash that involves the dermatomes that you should be knowing here. Okay, now just see here. This is the varicella zoster virus causing shingles in adults and chickenpox in children. Okay, it causes chickenpox in children. We had learned something that chickenpox, if it happens in pregnancy, especially in the early pregnancy. So first of all, pregnancy, if you start, one is early, one is near the delivery. Okay, near the delivery. The point is, early pregnancy में congenital varicella syndrome. Congenital varicella syndrome. This is what that would be seen. And the next one is this one over here. Near the delivery, if it is coming, it is going to manifest as neonatal varicella. Okay, it manifests as the neonatal varicella. Cool. Next, have a look here. Epstein-Barr virus. The next one in the list here is, known as Epstein-Barr virus. EBV, that is your Epstein-Barr virus. This is by HHV4. Epstein-Barr virus, it is by the HHV4. Kissing's disease and cancer, this is the word. Okay, Kissing's disease or the cancer, this is what is caused here. So get an idea, please. Kissing's disease, as we may have learned before, is caused by two guys, Epstein-Barr virus and cytomegalovirus. Epstein-Barr virus are, antigens are going to look like RBCs of sheep or horses. RBCs of sheep or the horses. And this is why it's called as heterophile test positive. When two antigens look same, it's called as heterophile antigens. And this is very rarely seen in 2 to 3 places only. One of them is right now. Paul Bunnell test positive is EBV. EBV. Okay? Cancers, since EBV affects the B lymphocytes, since it's affecting the B lymphocytes, what kind of cancers are expected? Lymphomas. Because of the lymphocytes word, you're going to expect the lymphomas. Done? That is the guy, Epstein-Barr virus. One thing it causes is Kissinger's disease. Second is cancers. Next, cytomegalovirus. Okay, next one in the list is what? Cytomegalovirus. Now, if you want to know something about this guy, CMV, cytomegalovirus, this would be it. It is the largest herpes virus, first of all. Okay, it is the largest herpes virus. Next thing, in immunocompromised patients, what is it going to do? See, it's one of the torch infections. You must know this already. Micro-related, more important point is, it causes CMV retinitis. CMV retinitis in HIV patients, and in the transplant patients, this is the most common infection. Okay, in the transplant patients, this is the most common infection. All right, I hope this is understood by all of you as such. Okay, now what's the characteristic thing that we see in here? Everybody must know that owl's eye inclusion bodies. What is this one described as? Owl's eye inclusion bodies. Now, this owl's eye inclusion bodies are seen in the nucleus. So, intranuclear inclusions, they are going to show the owl's eye appearance. Intranuclear inclusions will show this owl's eye appearance. See, not to be confused, I had told you this before. CMV, the inclusion bodies are seen in two places, cytoplasm also, nucleus also. दो जगह दिखती है. CMV inclusions are seen in cytoplasm as well as nucleus. But where is it going to look like owl's eye if they ask you? You should know in the nucleus. This is where it's going to look like that. Okay. Next part, HHV6 and HHV8. HHV6 causes sixth disease because the rash presents on sixth day of infection, which is preceded by 5 days of fever, known as roseola or exanthum subitum. You should know this other two names also. One is sixth disease, exanthum subitum or roseola, because of the rose color patches, because of the rose color patches here. HHV8, my dear friends, it is going to cause Kaposi's sarcoma. HHV8, it is going to cause what? Kaposi's sarcoma. That's the word right here for you. Kaposi's sarcoma, only in HIV patients. This is all going to happen in which patients? HIV patients. Okay, so that's about herpes family. Okay, now have a look at this. The next thing is HPV, human papilloma virus. Now, what you need to remember is this basic things here. There are three main serotypes, the subtypes that you need to know as such in this topic. The numbers from 1 to 4, okay, they are the ones to cause warts, right? Basically known as the verrucous lesions. Next, you have the numericals, that is 6, 11, that are going to cause the anogenital warts, condyloma acuminata, and then 16, 18 onwards, they are associated with cancer of cervix. Okay, so that's what you need to know. And for these guys who are going to cause the cancer, these strains, just know they have something in their structure. Basically, 16, 18 onwards, they carry something. The proteins, what we call as E6 and E7 proteins, that destroys, right? The E6 and the E7 proteins, these guys, they destroy the tumor suppressors called as P53 and RB. See, P53 and RB, they're good for us. They protect us from cancers. Remember, guardians of genome. And this is what is damaged by these guys. Okay. Hope it is clear to all of you. E7 damages the RB, E6 damages the P53. Okay. Now next, moving on. Pox virus, molluscum contagiosum. Make sure everybody knows this. This is the one that has the central depression as such. The characteristic identification of this, the central depression over here. Okay. So that is the molluscum contagiosum warts. Okay. The bodies that you get to see in this are Henderson-Patterson bodies or molluscum bodies. Okay. So HP bodies or molluscum bodies are seen in which one? Molluscum contagiosum. Next is parvovirus B19. Parvovirus B19, it is the, single-stranded, smallest naked DNA virus, right? Single-stranded is the important word because all the DNA viruses are double-stranded. DNA is always double-stranded. This is one exception, single-stranded DNA virus, right? Everybody knows the characteristic part of this, the slap cheek appearance, right? So what is this disease over here? Fifth disease, rash presence on fifth day, and that has the appearance of this slap cheek. So slap cheek appearance. Okay. Next, aplastic crisis is the rapid decrease in the RBCs due to the virus itself suppressing the RBC. And if somebody already has an existing anemia, they get to see this more easily. Okay. So aplastic crisis, that's the word here. Okay. Next, very, very important viruses, as you all know, that is hepatitis viruses, doctors. Now, what are the things to be remembered in the hepatitis viruses? Please know the points here. There are 5 types of hepatitis viruses, but only 2 of them are spread by the fecal-oral route. Okay, only these 2 are spread by fecal-oral route, that is A and E. A and E. Next, what you are supposed to know here is this. The roots of hepatitis B are similar to HIV, right? We should all know that the roots are pretty much same as the HIV. That's the thing that you need to keep in mind. Next, moving on here. If you see the word fulminancy or mortality, this is one that is expected. I'll just hammer that point in your head, which you may already know. Hepatitis E, E for elephant, is the one that causes death in pregnancy or its most severe infection if it happens in pregnancy, right? Fulminancy. That's the word. E is the one that is causing this. Now, talking about chronicity, if I'll ask you to choose one answer, who's the guy who has highest risk of becoming a chronic hepatitis infection? The answer to that is C. C for chronic. C for chronic. Okay. So 80 to 90% cases become chronic, whereas B also can become chronic. I'm not denying that, but only 10 to 20% cases can become chronic in here. Okay. So that's the word. So because these guys can become chronic, they can actually manifest as cirrhosis, etc. Hepatocellular carcinoma, cirrhosis, etc. This is what is going to happen here. Okay. Next. Moving to this guy, just have a look here. Hepatitis B. Hepatitis B. These are the 3 antigens, HBs, HBe, HBc. Okay. HBs, HBe, and HBc. These are the 3 things that you should see here, first of all. Now, if you understood what I just mentioned, 3 names here, you must have recognized each one of them has something to say. HBs, if you are able to find in the patient's sample, let's say you took a sample, our test detects different things. HBs antigen, if you find in someone, you should say the person is having the disease right now. Okay, if this antigen is found, that means we should be saying the person is having disease right now. Next is HBe antigen. Remember it for infectious nature of the virus. If this antigen is present, the person is highly infectious. The person is highly infectious. Okay, if this antigen is present. And this core antigen is there in the virus, but our test cannot detect it. Our test cannot detect it. Okay, so I'm going to remove this thing from here now. So what are the things you will see in the report if I ask you? HBs antigen and HBe antigen. HBs antigen means disease is present. HBe antigen, if I tell you, the disease is present along with high infectivity. That's what you need to know. Now, if you look at the antibodies here, the point is here that is three antibodies can form anti-HBs, anti-HBe, and anti-HBc. See, I know you will say, sir, you said the HBc antigen is not detected. Yes, HBc is not detected by our test, but it's there in the virus. Now, our body can detect this antigen and make antibody. So antibodies are present in the body. So now understand this point. What is the meaning of seeing each one of them? This antibody, anti-HBs, tells you that you have gained ability to fight this infection, that you will not have hepatitis B after this. Okay, anti-HBs if you have developed, that means you have gained ability to fight this virus. Is that clear enough, doctors? Anti-HBe means less infectious nature or no infectious nature. That means this guy has lost the ability to spread the disease as much. And this one, anti-HBc, the yellow one, can come in two flavors, IgM and IgG. If IgM you see, you should say acute infection. IgG you see, you will say chronic infection. So is that understood by all of you doctors? IgM and IgG, that's the word that you need to know as such. Done. Now look at the vaccine strain. In the vaccine strain, when I am injecting you with the vaccine of hepatitis B, only one antigen is present in that. That is HBs antigen. Okay, that is the HBs antigen. Okay. Now the story is this. If your body is exposed to only one antigen, it will make only one antibody. That means in a vaccinated person in some time, right? What is the thing that is going to happen, doctors? Only one antibody would be seen called as anti-HBs. So technically, anti-HBs antibody, the red antibody, can be seen only in two people. Either somebody who previously was infected, had a disease before, and the second one is this one. Okay, second one is this one, vaccinated person. Okay, so this is something for you all to know. HBs antigen, it is called by the name of Australian antigen, as you can see. Okay, it's the first marker to rise in the blood. Okay, please do remember that one. Have a look at this, try and identify. Just I'm going to do one or two examples for a refreshment. See here. Always look at the HBs antigen first. Confirm if the disease is present or not. If you see HBs antigen, you know the disease is present. HBs antigen, disease is present. Anti-HBc, if you see IgM type, anti-HBc, if it is IgM type, you know it is acute hepatitis B. And second, if you're seeing HBe antigen now, you should all know this is infectivity, high infectivity. Acute hepatitis B with high infectivity. This is the word that you should all know. Okay, so that's about it. Next, HIV virus. First words first, the most common HIV in India, we have used the mnemonic MC only. The most common is MC, right? Group M, subtype C. Group M, subtype C. This is the word that you need to remember, group M and the subtype C. Okay. Next, HIV virus, the structure is based on three genes, ENV gene, Pol gene, and the GAG gene. Okay, ENV gene, Pol gene, and then the GAG gene. These are the three genes that you're looking at right now over here. Okay, next thing. What are you supposed to know is ENV gene codes for the envelope proteins like GP120 and GP41. So GP120 and GP41, this is what you need to know as such. GP120 and then GP41. Okay. Next, Pol gene codes for the polymerases. It codes for the polymerases. Example, reverse transcriptase. Okay. GAG gene codes for P24. Take it. Now, I'm just going to show you something. When the virus comes inside, see, number one, what are the targets? HIV entered the human body. This virus, HIV virus, now the target cell is T helper cell, the boss of our immunity, because it wants to suppress the immunity, right? So for this, it should bind to the receptors on the cell called as CD4, CXCR4, and CCR5. Do you all remember these names? CD4, CXCR4, CCR5. That is where the target of HIV is. Once it comes inside, I told you it takes out three enzymes one after other: reverse transcriptase, integrase, and protease. Reverse transcriptase, integrase, and then the protease. These are the three enzymes that you should all know. Okay, समझ आया सबको? So that's your three things: reverse transcriptase, integrase, and then the protease. ठीक है. Now let's move forward here. Very important thing is, how long does it take for this guy who has developed the infection to develop into AIDS? See, HIV-1, you just remember like this: HIV-1 takes 10 years, HIV-2 takes 20 years to develop into AIDS, right? See, nobody wants to develop into AIDS faster, right? So this guy is said to be more dangerous, that's why. Okay, so HIV-1 is considered to be more dangerous. Okay, is that understood, doctors? Now, if you should know the normal CD4 count in a human being, okay, it is less than, normal CD4 count, it is said to be 500 to 1500, which every one of us has learned. So anything that is less than 500, now you have become immunodeficient, right? Now, in this also there are levels: less than 500, less than 200, etc. Now, the point is, less than 500, we can expect the people to become this immunodeficient due to various causes, but less than 200 can happen more or less due to HIV. Okay, that's why some specialized infections which are seen only in HIV are seen here. Less than 200. Are you understanding what I'm saying here? Less than 200. This is the CD4 count where you see this kind of things. Less than 200. Less than 200. And what were those things? You already have learned. One fungus causing pneumonia called as pneumocystis, and one fungus causing meningitis called as cryptococcus, right? Less than 500, or overall most common infection, in HIV patients, it is tuberculosis, doctors. Okay, it is tuberculosis. Most common infection that you should be knowing is tuberculosis. Okay, so that's about that. Next, you can see here the diagnostic aspects now. Okay, when somebody is exposed to HIV, okay, how early I can detect something? The answer to this is roughly by day 12. Roughly by day 12, I can detect the first thing, that is HIV RNA, by doing the PCR, right? The earliest diagnosis test I can do, right? It is the RNA, HIV RNA, okay, by PCR. Is that clear enough to all of you? This is the first thing. Now, few days later, that is by day 16 onwards, another antigen starts to appear, that is P24 antigen. P24 antigen. P24 antigen, it is an antigen of HIV virus. Okay. So this antigen test, P24 antigen test, This is the one I would want you to answer. If the question asks you recent history of exposure or window period, recent history of exposure or window period, what is the test to be done? I mark my words, you should answer P24 antigen. The best test is PCR, I agree, but we do which one? P24. Okay, so please know this thing. Okay, for answering questions, we are not talking about just the knowledge right now. To answer the MCQ, what is your answer? P24 antigen. Okay. Next, antibodies take time to appear. They take roughly 3 weeks to 3 months. So it's a long, wait for that to form. Antibodies take a longer time to form as such. So that's the thing. So I'm just revising few MCQs for you. If the question asks you recent history of exposure, then you're answering P24. If they ask you window period, you're asking P24. If they ask you most sensitive test, you're answering ELISA. If they ask you confirmatory test, you're answering Western blot. I hope there is no more confusion after this. Recent history of exposure, please mark P24. Most sensitive, ELISA. Confirmatory in India, Western blot. Best test overall, it is PCR or NAT. But according to the guidelines, we are not going to do that. According to the guidelines, you have to choose Western blot. Is that clear enough to all of you? So these are the 4 words in relation to the HIV virus. Okay. Next, you might remember influenza virus. Influenza virus is the one that causes the bird flu, swine flu, etc. Influenza virus is the one to cause the flu. Okay. Next, influenza virus has very characteristic number of pieces of RNA. It has 8 pieces of RNA. Okay, it has 8 pieces of RNA as such. Okay, so this is what that you should know. Next, look at the word HNN. When we say bird flu and swine flu, we use the word H1N1, H5N1 like that. Yes or no? So what you are supposed to all remember is the H and N are antigens of this virus. H stands for hemagglutinin, N stands for neuraminidase. H mediates entry into the cell, N mediate exits from the cell. Okay, so that's the thing. H and N, this is the story. Now, next part, see here. This is important. They can ask you about the antigenic variation. There are 2 things antigenic variation wise, antigenic drift and antigenic shift. Antigenic drift and then the antigenic shift. Please know drift is a smaller change. It's a minor change in the antigenicity and it's happened due to a change only in one place called as a mutation, point mutation. Whereas antigenic shift happens due to genetic reassortment. Okay, genetic reassortment. This is the word right here. All right. So one is a minor change, one is a major change as such. Okay. Next, moving on, it is going to present as a respiratory tract infection, just like the COVID. Okay, it's going to present as a respiratory tract infection, just like the COVID. And this is what I would want you to know, doctors. Please see here the reference that I'm trying to give you. The target receptors are going to be different over here. Okay, so have a look. First thing is this. Okay, if we are talking about influenza virus, the structure called as hemagglutinin on the virus binds to the respiratory cell by the receptor called as sialic acid receptor. So our respiratory cells, what is the receptor here? It is sialic acid receptor. And who's binding over here? Hemagglutinin. Hemagglutinin, it is binding over here. Okay, so that's how the infection happens here. Next, if I go into the other one, okay, see here, the next one here is this COVID. If you talk about COVID, the virus, coronavirus, has something on its surface known as spike protein, we have learned. Do you remember this word? Spike protein. And guys, this will bind, spike protein, it will bind over here to this thing. Which one will it bind to? Spike protein. It will bind to the receptor called as ACE2 receptors. It is going to bind to the ACE2 receptors. Apart from this, there's nothing much I need to tell you as such in the COVID also. Please just remember when we do the PCR, right? RT-PCR, there was one value I had told you called as CT value. Okay. There was something by the name of CT value. So what is this CT value? Please understand the point. This is not the CT score, what you see in, the HRCT, not the radiological one. This is the microbiology report, PCR value called as CT value. I said the cutoff value is 34. If somebody's value comes out to be more than 34, that is 35, 36 onwards, this person is said to be COVID negative. Any value less than 34 is considered to be positive. So less than 34 is positive, more than 34 is negative. So what we are saying is that 34 number, how did you come? That means numbers 1, 2, 3, 4, 10, 20, 30, 34, like that. All these numbers have been there. So the cycles that were done, CT stands for the cyclic threshold, So number of cycles that were done here are 34 cycles. And if you're able to find the virus in the early cycles itself, that means there is so much virus in the body. And if you're able to, find the virus here at the 30 cycle, that means it's very less virus. Why? Because 34 is like negative. This is the negative value. And if your testing value is very near to the negative, that means this person is having very less, virus inside their body. So that's about that we are going to do in the COVID, nothing more that I'm going to tell you as such. Okay, so that being said, let's move on. Paramyxoviruses. Now, these are just plain and one-liner organisms. Okay, para-influenza viruses, croup, right? So you should know this thing, that is the croup. Croup causes what? Laryngotracheobronchitis. Laryngotracheobronchitis. This is the one that is appearing as the barking cough. Okay, it appears as the barking cough. So this is what you should know. Next, mumps virus. It causes parotitis. Okay, the inflammations it causes is parotitis, inflammation of the parotid glands leading to the swelling here. Next, orchitis. Orchitis, this is the other thing as such that you should know, right? Testis, ovaries, etc. All this can be swollen basically by the mumps virus. Okay. Measles or rubiola. Measles, you have to know this proper sequence, doctors. If you're going by the incubation period, first 10 days, it's almost silent. 10 days, the fever. 12th day, there is appearance of Koplik spots. 14th day, there is appearance of rash. If you minus the incubation periods and talk directly, after the fever, 2 days later, Koplik spots will present. 4 days later, the rash will present. Okay, this is the sequence. And now what you are all supposed to remember is, along with the fever, the other things that we get to see are the 3 C's of measles. What were the 3 C's of measles? Cough, coryza, conjunctivitis. Okay, so what are the 3 C's here? Cough, coryza, and then conjunctivitis. These are the 3 C's of measles. Okay, I hope this is clear enough to all of you. 3 C's of measles are cough, coryza, and conjunctivitis. Okay, done. Next, Moving on, a very important complication that they might ask you here, SSPE, subsclerosing panencephalitis. SSPE, this is a complication of measles, subsclerosing panencephalitis. Now, when you take up the diagnostic part of this, you get to see this cells called as Warthin-Finkeldy cells. Yeah, this in the measles, we see this macrophages by a fancy name, Warthin-Finkeldy cells, Warthin-Finkeldy cells as such. So that's it. That's about this guy. Next, rubella. Just read that the rubella is important for congenital disease. What does the rubella do? It is three things, what you're seeing here, that is blindness due to cataracts, etc. Heart disease like PDA, patent ductus arteriosus, and then deafness. Okay. Now, arbovirus. Next virus in this list is which one? Arbovirus. Now, what are you supposed to know with the arbovirus? Arthropod-borne viruses. Okay. Arthropod-borne viruses, meaning some insects are involved in spreading these diseases. Okay. Some insects are involved in spreading these disorders. So if you have gotten this idea, dengue fever virus, first one. Okay. Dengue fever virus. Please know this guy. It has multiple serotypes that you're seeing here, DEN 1 to DEN 5. Okay. Transmission of this, it is done by this mosquito called as Aedes mosquito. Do memorize the vector-borne diseases. Who's the vector? It would be important for you all. Okay. Next, what is the specialty as such in the disease? Let's try and understand. The primary infection would be caused by one of the serotypes. Okay, one of the serotypes is going to do this primary infection, and secondary infection is going to happen after the primary, of course. So this is the first time you were exposed to dengue virus. This is the second time you're exposed to dengue virus. See, the catch here is you should be getting infected with different serotypes here, not the same one. You should be getting infected with the different serotypes. Is that clear enough? Okay. Now, try and understand this. Antibody dependent enhancement. Antibody dependent enhancement. This is what you need to know here. This is a very important thing. So what is the story? First time you got infected, see example is written here. We'll take the same example. Let's say the first time you got infected was by the first serotype and antibodies were made against it. You will not get infected by DEN 1 again because antibodies has been made already. Suppose later in life, you got infected with another serotype DEN 2. Now, these antibodies, of course, they will not protect you from this because it's a different serotype, right? But what are these antibodies now doing is that these antibodies are actually going and, protecting this virus. Our own antibodies, instead of fighting infection, they are actually protecting this virus. Because they are protecting the virus, what ends up happening, doctors, serious disorder will happen, that is DHF and DSS, dengue hemorrhagic fever and dengue shock syndrome. Okay, dengue hemorrhagic fever and then dengue shock syndrome. This is the thing that you need to know here. Okay, so what is seen here is this hemorrhages because of, platelets getting less than 1 lakh, hemorrhages would be seen. And if you don't control this bleeding right now, it will lead to shock. It will lead to shock. Important micro questions, what they can ask you, this one. How to diagnose this? Please know, first week, obviously, any disease, antibodies will never be formed. At least it takes 1 week for antibodies to be formed. Okay, at least it takes 1 week for antibodies to be formed. So is that understood by all of you? At least 1 week, remember. So that's why in the first week, what we are going to do is, if you want to diagnose, you should look for the NS1 antigen. You should look for what? NS1 antigen. It's one antigen of this virus. Just look for that. And 5 days later, the first antibody to be appearing is IgM as usual. So IgM will be detected. Okay, so that's about that. I hope everybody understood this thing. Next, moving to other things in here. Japanese encephalitis, may please remember it is the most common cause of encephalitis. Okay, and the mosquito here is Culex mosquito. Culex mosquito. Okay. Very lesser yield, don't pay attention to that KFD, etc. Next, rabies virus, very, very high yield, definitely important for your exam. Okay. Next, you have your rabies virus. Now, just look here, doctors. When we are saying the rabies virus, rhabdoviridae, what do you all need to know is this virus, it's a bullet-shaped virus. Okay. It is basically a bullet-shaped virus. So this is what that you need to know as such. It's a bullet-shaped virus. Done. Next, structure-wise, okay. There are two parts, glycoprotein G and nucleoprotein. There are two antigens you are seeing here. One is glycoprotein G and the second is what? Nucleoprotein. Okay. Now, understand this. Glycoprotein G, this is the guy that plays an important role in pathogenesis. In other words, it helps in causing rabies. How does it help in causing rabies? It binds to the receptors, what we call as acetylcholine receptors. Okay. So technically, this guy, glycoprotein G on the virus, it's helping it to bind to the acetylcholine receptor. And from here, at the fixed speed of 3 mm per hour, the main target is brain. So slowly the virus is going to reach the brain through the nerves. And this is known as the retrograde transport. Okay, retrograde axonal transport. That's the word that you should all be familiar with over here. Okay, so that's about this guy. चलो अभी यहाँ पर आते हैं important thing. How do you get this disease? One is obviously bites from the rabid animals and licks on the cuts, right? Cuts known as abrasion. Once this disease presents, you know it presents with this classical features like hydrophobia, etc. A very important parts are the bottom two, diagnosis and the management. See, if the person is living, we are going to do the diagnosis by collecting samples from the backside of the neck. And what are we going to collect here? Hair follicles. We're going to collect the hair follicles over here, right? I hope this is understood by all of you. Next, once you have taken this, there is a test called as direct fluorescent antigen test called as DFAT that is supposed to be done, which detects the nucleoprotein antigen, or PCR can be done. Okay, so DFAT is direct fluorescent antigen test. As the name says, we are detecting an antigen. What is the name of the antigen? Nucleoprotein antigen. And then PCR is another test. So if the person passed away due to rabies, suspected case of rabies, or, let's say a death, which is suspicious death. I don't know, the person died, young person. So I'm suspecting it can be rabies because I'm seeing, some marks on the body, bite marks. So if suspected case of rabies, now what we can do is Negri bodies have to be, looked for. Negri bodies are seen in brain in three parts: hippocampus, cerebellum, and brainstem. And for the Negri bodies, you need to use a stain called as Seller's stain. Okay, the stain to be used here is which one? Seller's stain. Please do remember this keyword right now. Okay, now that you must have seen this, let's look at the management. I'm just going to tell you the category 2 and 3. Category 1 is super simple. There is no risk of rabies. No treatment is needed. That's it. No risk of rabies. No treatment is needed also. Okay. Now, next is this. If you're talking about, okay, minor risk versus major risk, what to be done? First, you should know how to identify it. Who's called as a minor risk? Minor abrasions, small scratches by the animal on your body, which are not bleeding. If there is a scratch which is not bleeding, you should know it's a superficial scratch. Then the risk of rabies is also less. Again, risk is less, but it's not completely absent, right? So you're not going to take it so lightly also. So what to do in the safe side? First, local treatment. Clean the wound properly, right? Wash the wound with abundant of, soap and water. Clean that wound with antiseptics like betadine, etc. We can use those things. And then give rabies vaccine to this person. Give rabies vaccine to this person. Okay. Next, category 3 is the major risk guy. You're seeing animal bite with a bleeding. You're seeing licks on abrasion, whatever it is in the history, bite by wild animals, etc. This is all called as what? Major risk. And guys, here's where you have to do something. Local treatment as usual, antibodies and vaccine. Antibodies as well as vaccine. All this needs to be done. So what is added here? Ready-made antibodies called as HRIG. Okay, ready-made antibodies by the name of HRIG, human rabies immunoglobulin. Okay, that's it. Next, in this picornaviruses, I'm just going to tell you this. Please keep an eye on this one, hand, foot, mouth disease. Important. Hand, foot, mouth disease is caused by two viruses, enterovirus 71 and Coxsackie A16. Okay, enterovirus 71 and Coxsackie A16, they are going to cause this thing, hand, foot, mouth disease. Coxsackie B for B, please remember the name of the disease. That's how they can ask questions, even though it might be a rare thing in reality, but they might love to ask that. Bornholm disease. What is the name of the disease here? Bornholm disease. Okay. This is the guy that is going to be known as devil's grip because it is a myalgia in the intercostal muscles. Okay. It's as if somebody is gripping over you. That is the word. Okay. Now, COVID, etc., we will not talk because there's barely anything. As I told you, what you needed to know as such before only. ACE2 receptors are the target receptors by COVID. And what is the main guy in the COVID that's doing the disease? Spike protein. Spike protein. Okay? So now this is the Ebola virus. Very less likely to be asked. Again, just the picture I'm showing you. This picture should be in your head if they ask you, this is Ebola virus. Okay, so that's your virology section, guys. Next is the bacteriology part. Okay, next one, it is the bacteriology part. Now, just for a reference to start this topic, I'm going to tell you these two things that you should all know. Okay. First thing, if I am saying gram stain, if you see purple color versus pink color, who is who? Okay, that's what we need to know. Purple versus the pink color. Next, in the acid fast stain, who is who as usual? That's what we need to know. So gram stain, purple color organisms, as you may all remember, are gram positives. Gram negatives are pink in color. In the acid fast stain, acid fast are pink in color, and non-acid fast are blue in color. Okay, non-acid fast, they would be blue in color. I hope this is understood by all of you. Okay, gram stain and then the acid fast stain. Now that this is over, please try and understand these things. Gram positive or gram negative, they are further divided into two guys, cocci or bacilli. Cocci are the round, bacilli are the rod-shaped organism. Who are the cocci which are purple in color? If you see purple in color, you should know either it is staph or either it is strep. If you see the round button pink in color, you should know it is Neisseria. You should know it is Neisseria. Done. Next, we have the rod-shaped organisms. You should be memorizing it as ABCDLMN. ABCDLMN. These are the rod-shaped organisms. ABCDLMN. Okay? ABCDLMN. Rod-shaped organisms. Everybody else, it is gram-negative rod. Okay? So that's about it. The rod-shaped purple, if you see, you should know we are talking ABCDLMN here. Now, who's in the ABCDLMN? Actinomyces, Bacillus, Clostridium, diphtheria, Listeria, Mycobacterium, Nocardia. That's it. These are the guys that are called as, gram positive rods. Gram positive rods. ABCDLMN. ABCDLMN. Okay. Now we will start here with this first guy. Gram positive cocci. Either it is staph or strep. How to confirm if it's staph or strep? The biochemical test is catalase test. You all know if you see catalase test, you have to look for the bubbles. You have to look for the bubbles. So catalase positive organism is staph. Catalase negative, it is strep. So catalase positive would be staph. Catalase negative would be strep. So that's the word that you should be knowing here, right? Staph versus strep. So if you see this, you know it is staph. If you don't see this, then that is strep. Done. Once you have confirmed whether they are staph or strep, now the next thing comes to know which species are they. Which species are they? So for this, I would want you to know. Let's assume you saw bubbles, staph. Now here we are, Coagulase test. Next one in the list is Coagulase test, where you're going to see for clumps or clots. Okay. We're going to see for what? Clumps or clots. Coagulase test. Basically here you have a bacterial suspension along with plasma. So you have to use, plasma, that is blood, and add the organism into that. After adding the organism, if it's forming clumps inside, that means clotting is taking place. So as simple as that, that organism who can clot, it's called as coagulase positive. The enzyme coagulase will lead to clot formation. If you see that, the organism is confirmed to be aureus. And coagulase negative, it is said to be cons. And what is the cons? Coagulase negative staphylococci. Coagulase negative staphylococci. So that's the word here. Okay. If you don't see clumps, you probably will recall this picture that all of us have seen that time. This is a picture showing your, antibiotic testing. Okay. Pay attention. After adding antibiotic, if you don't see any change around the antibiotic, as you see the cross marks, there is no change around the antibiotic here. That means your antibiotic is not working and you should call this as resistant. That means organisms are resistant to this antibiotic. Whereas after adding antibiotic, if you're seeing changes around the antibiotic, as you can see by the arrows, you're seeing some area getting cleared as such. This is known as zone of inhibition. Okay, what is the name over here? It's called by the name of zone of inhibition. Okay, I hope this is okay with all of you. So this is sensitive antibiotic. It is killing the organisms, right? It is killing the organisms. So this is zone of inhibition. Done. Next. So this is coagulase test. If you see this, this is aureus. This is the cause, coagulase negative staphylococci. And then you should know this is the story right here. Okay. Next. This picture, quite easy. Everybody should know this. If you see the cocci, purple color cocci in clusters, it is considered to be staph. If you see this rather in chains like this, then you should say it is streptococci. Okay. In chains, it is streptococci, and in clusters, it is this. Okay. So that's about that. Now, let's go back to this topic. Once you have confirmed who is who, a reminder to all of you, one word. What is the disease by saprophyticus? UTI. It is a UTI causing agent in young females, but it's not the most common cause. Okay, it can cause the disease in young female. You have said it enough, but it's not the most common cause. That is first thing you should know. Next, staph epidermidis. Staph epidermidis. This is the next guy. Please remember, staph epidermidis, it causes endocarditis. It is the infection of the valves. Okay, it's the infection of the valves. It causes what? Endocarditis. Okay, if you understood this, that is causing endocarditis, please just remember one thing. Endocarditis these days is divided into two words primarily: native valve endocarditis, that's the original valve, and then the artificial valve known as prosthetic valve. And when you have implanted prosthetic valves in someone's heart, expect infection of that to come called as endocarditis, which is divided as early onset and late onset. Do you remember? Prosthetic valve endocarditis is divided as early and the late onset. Early onset is E for epidermidis. Late onset is by viridins group like sanguis. Example is sanguis, although there are others that can do this. Okay. Next, that's about that. Staph aureus superstar causes thousands of infection, as you can see here. Pretty much the diseases are divided into two types. Aureus diseases are divided into two types: organism mediated and toxin mediated. Organism mediated and then the toxin mediated diseases. These are the ones that you should be knowing as such. Okay. Organism mediated and then the toxin mediated diseases. Okay. Now, watch the things here. When I'm saying this word, toxin mediated, there are three main diseases: food poisoning, toxic shock syndrome, staphylococcal scalded syndrome. When we say food poisoning of Staph aureus, you should know it's a superstar. The toxin will not get destabilized by heating the food. So it's a heat stable toxin. And the symptoms is fastest onset. So we can say heat stable and then fastest onset. This is the word that you all should understand. Okay. Now that being said, now look here. Toxic shock syndrome is going to present as shock. Toxic shock syndrome is going to present as shock. Okay. So that's about this thing right here.